So if you have any questions, please feel free to drop them in the Q&A as we go, and we'll get to them at the end. But without further ado, I would like to now hand the floor over to Katrin. Thank you very much.
So hello, everyone. Thank you, Ben, for your introduction. Just also to summarize shortly what we will talk about, as Ben already said, we want to give you a short introduction to who we are, who is SimCell. Then you will learn the basics, very limited basics about biocalorimetry. And also I will give examples of applications for probiotics analysis. And then I hope there will be nice and interesting questions from your side. Calorimetry basically means measuring heat. And I don't know how many of you are familiar with calorimetry, and especially I think not so many will be familiar with calorimetry and applications in biology. When I studied biology 17 years ago, or started to study, one of the first labs we had was actually physical chemistry. And we had to do an experiment with a device that is shown here on the picture. It is a calorimeter, and we had to add substances and then manually note down the temperature at given time points. At that moment, I thought this is a very laborsome technology, and also I found it complicated and thought I will probably never use it again for my former research in biology. Obviously, I was wrong. And at the same year when I entered university, SimCell was found here in Sweden. We are based in Stockholm on the KI campus. And it was found by microcalorimetry experts that actually had the idea that microcalorimetry is an excellent method to use in biology. The only thing that was missing at the time was a suitable instruments. So the goal was to build an instrument that fits the needs of a biology lab, basically a very sensitive instrument that allows multiplexing, that allows automated and continuous measurement, and also that fits the sample size, that you don't need to use excessive sample of your bacteria or any biological sample that you have. In 2014, the calScreener was CE marked, and also the first publication came out. And that was also when the company shifted gears. It became involved in a Horizon 2020 project that was pretty successful on rapid antibiotic susceptibility testing, but also at the time, we broadened our customer base, and we could prove that we have a lot of other applications in biology where we can bring interesting results, ranging from biofilm, but also infection models, cancer, adipose tissue research, just to name a few. But today, that is not our focus. We want to talk about probiotics analysis and development. But before we do so, I want to explain why you can actually use microcalorimetry in microbiology, and also why you actually should do so. And the basis of it lays in the bacterial metabolism. So bacterial metabolism consists of catabolism and anabolism. It goes for energy harvest, energy storage, and a lot of energy is turned over during these processes. And these processes are actually metabolic pathways that consist of individual biochemical reactions. And each biochemical reaction will release energy, free energy in form of heat. And that is basically what you can measure with calorimetry, with isothermal calorimetry. And how you can do that is shown here in a simple principle of how we measure it with a calScreener. So you can add any sample that that you can imagine in this reaction vial depicted here. This vial goes on a 48-well plate that will then be inserted in the calScreener. That is the calScreener from the side cut open. So you insert a blade and it will end on a, each vial will end on a heat sensor. And then basically what happens in your vial, so here an example of a bacterial culture. The bacterial culture will be consisting of many different cells that are metabolically active, and eventually they will produce heat, and this heat will be sensed by the heat sensor as heat flow in microwatts. And what we can say is that the heat production rate is directly proportional to the metabolic rate of the sample. And what you then get is a thermogram, which is basically the heat flow over time. And now, why should you then use it for probiotics development and analysis? So the probiotic life cycle consists of many different steps, as you know, and that goes from strain selection to process optimization quality, but also shelf life and stress tolerance. In all these different steps, you might lose amount of viable cells, or they will shift into an inactive state. And the thing is that in the end, you want a probiotic product that has as many viable cells as possible, and also actively viable cells. The problem right now is that we lack methods that can actually measure viability in a fast and easy way. And isothermal calorimetry is a method that can, in a simple way, help you to do a viability assay, but also easily detect contaminations in your sample simply by a metabolic activity readout. And that is shown here on an example. So you imagine you have probiotic strain and you want to upscale and produce it, but therefore you want to test where the strain is having the maximum viability and vitability. And then here on this example, we tested 3 different media, MRS, like a standard medium, and 2 home formulations. And the goal here was to check the maximum activity and performance of the strain. So the different colors depict the 3 different media, and on the left side of the graph, you see here on these lines the heat flow curve, and on the dashed line that is pH curves. And you can see when in medium 4, in broth 4, when the pH drops early, also the metabolic activity will reach a much lower peak as when you have a pH drop much later in the growth process, where the maximum metabolic activity reaches much higher. So you can look at the performance of the strains directly with a metabolic activity readout. But when you look at simply biomass formation, which is shown on the right example, where we measured with flow cytometry the individual cell counts, this is not depicted. There is no difference in the 3 different media in biomass formation, but clearly there's a difference in performance. So what you clearly get with isothermal calorimetry is you get sensitive metabolic data that are far beyond any growth assay. The signals that you get are robust and sensitive and allow a rapid detection and enumeration of viable cells. And that is shown here on the example that is E. coli cultured in rich medium with different inoculum sizes. And what you can see that the time to detection, the lower the inoculum is shifts to the right on the time scale. And it has a direct correlation of detection time with the inoculum size. So it is an excellent method to enumerate viable microbial cells. And you simply have to add the sample directly to the incubation vial. You can use any strain, any medium, as here is an example of different strains, E. coli, Enterococcus strains, Salmonella, grown in different media, for instance, Müller-Hinton, Glatt, broth, but also milk. What you need is a standard curve for the strain and the specific medium, and then you can have a very fast detection of the cells and a fast answer how much viable cells were in the original sample. The thermogram is also species and strain specific. So you obtain something what we can call a metabolic fingerprint. And that are two different examples shown here. On the left, lactic acid bacteria grown in plant milk. And you can see that all three strains that were tested here show a different thermogram. And the same is on the example on the right, also different Lactococcus lactis strains that were grown in plant milk. And you can see even on the strain level, you get different thermograms. So you can see that this is a very sensitive measurement of the metabolic activity, and we can use that fact also to assess the substrate quality on the and get a metabolic output. This is an example of E. coli grown in minimal medium with different additions of glucose ranging from 0.1% to 2% glucose. And you can see that even the small changes in glucose addition will change the thermogram. So this gives you the opportunity also for substrate batch control to, for instance, assess the quality of a certain substrate. For instance, if there's degradation or impurities, you could eventually see that with this sort of assay. But also it allows you to compare media formulations to, for instance, improve the process of your product. And again, nothing is hidden from biocalorimetry, so this method can also help you to easily and fast detect microbial contaminations. So again, on the basis of that, you have a metabolic fingerprint that is unique to the species and the medium condition. You can easily detect that, and that is shown here on the graph. So you have a unique thermogram for a lactic acid bacteria, and also a unique thermogram of S. aureus. Both here were grown in milk, but if you co-cultivate them, you can see that the thermogram changes. So if you cultivate your known bacterium, you exactly know how the thermogram looks, and if there's a change, that would clearly indicate that you have a contamination in your batch. To summarize here, you get 4 quality parameters in one assay. So first of all, it is a very robust method to estimate the microbial content. Also, you get a strain-specific metabolic profile, and also direct detection of contaminant microorganisms is possible. Another interesting and exciting thing is that you can have a phenotypic assessment of the metabolic health of your strain, but also you can directly assess the quality of your growth medium. And now I will show you a few more examples what you can eventually do with isothermal calorimetry. For instance, you can do co-cultivations and test for bacterial interactions in a very easy way. And here is an example for strict anaerobic bacteria that were isolated from the gut. And when grown in monoculture, you can see here the thermograms of one and the other species in blue. But when they were combined, the species, the time to detection shifted to a much earlier point, and also the metabolic activity increased. But when taken endpoint CFU count, there were hardly any changes in CFU after 24 hours cultivation. And that was a very interesting observation to see. So there's certainly some synergy or another effect going on in the co-cultivation of these two strains. And since the method is non-invasive and non-destructive, you could in the end use the sample, open the vial, take it out, and do additional experiments to investigate what was going on in the sample. Another interesting thing what you can do with the method is to assess the stress tolerance. For instance, when you ingest probiotic, it needs to pass the GI tract, and this type of stresses also that the microorganisms face. You want to see how much cells actually survive or stay viable throughout the process. This is another example for stress. This is dehydration stress, but it also just simply to illustrate how you can assess stress with this method. So here, bacteria, S. aureus in that case, was put on a PVC membrane and dehydrated for 168 hours. And then for the measurement, you could add the PVC membranes directly to the vial at medium, and then record the thermograms. And since, as I already told you before, you can use the method to determine the initial cell numbers, we could see the reduction of the viable cells due to the hydration. You're not restricted to look at simply monocultures or co-cultivation experiments. You could also add very complex microbial communities in the vial. And this is an example of sludge isolated from lake sediment. And that was then exposed to DCP, a herbicide with different concentrations. And you can see that the microbial activity, so this is the untreated sludge, the microbial activity of the treated sludge with DCP went down. And when you remove the pressure, the chemical pressure of DCP, and regrow the samples, you get thermograms, but the thermograms change for the different concentrations, and that indicates community switch in the sample. So this is another way to access. Also, you could imagine to test different prebiotics, but also probiotic strain on the activity of a complex microbial sample. So this was the examples on the probiotics. I hope that was interesting for you. We will now take some questions, but also you can connect with us for more information.
Thank you very much indeed, Katrin. And yeah, if anyone has to leave now, I know some people said they couldn't stay the full time, then please take a note of Katrin's email there. You can reply directly to the email that you've got for the webinar, and we'd be happy to send you any extra information, like we have the publications and stuff in there that you might be interested in reading. So please feel free to reach out to us and to ask us any questions right now, if you like. There's a Q&A button at the bottom here in Zoom, and there's also a chat, and you can also reply to the emails. I'll be checking all of them now, so you can see if anyone has any direct questions. To start with, and this is a question to you, Katrin, but it also might be for people in the audience, because I know we have a lot of experts in probiotics there, but what are the most, what are the probiotic products that could probably most benefit from this, meaning that the kind of methods that are there now are a little bit lacking and it might take too long, or it might be difficult if you're dealing with kind of solid or opaque samples? Oh, I can't hear you there, Katrin. Is your microphone?
Yeah, can you repeat then?
Yeah.
Yeah.
So I was just wondering, like, what kind of probiotic products do you think would most benefit from something like biocalorimetry, like a new method? You know, where do you think kind of current methods are lacking? Or if, I mean, it's difficult for us to know the probiotic products, which is why people in the audience might help. But like, what would the attributes of a product like that be that would be specifically, you know, kind of well placed to take advantage of biocalorimetry?
I think in principle, any probiotic product can benefit from this technology simply because the current methods, I know people use a lot flow cytometry, but also quantitative PCR techniques. And I think we can also be relevant in that field and even give additional information to these techniques that we mentioned. So it can be also life therapeutics. So anything that deals basically with microbes, and that is the products that actually probiotics are. Yeah,
I see. No, that's it's really interesting to kind of hear the feedback from people in this area, because it's quite a good fit, really. I mean, yeah, let me just have a look at the Q&A now in the chat. Has anyone got any questions for us? I'm looking in the audience there now, and I see some names I recognize. I even see some people that we actually cited there in the presentation, which is nice to see. So we've got some experts in the audience, which is nice. So yeah, any kinds of questions that you want to ask, it can be kind of detailed questions or kind of basic questions on the technology. They're very, very welcome indeed. I'll just check I haven't missed anything there. No. Okay. So yeah, in terms of replacing current method, you kind of mentioned flow cytometry. Is that the main thing that biocalorimetry could potentially replace, or are there other methods, other technologies you think that we could actually do a better job than?
I think the advantage of microcalorimetry is that you don't distract the sample. So as compared to qPCR, you leave your sample intact, and you can always add downstream experiments to it, the same as for probably, well, in flow cytometry also you will not kill the cells, but still it is a very cost efficient method. You are independent from adding dyes to your sample, which you might want to use when you do flow cytometry experiments. So I think there is a clear advantage in that sense of the technology, that you are completely independent from reagents. You simply use your cells and the medium.
Yeah, I mean, it is pretty simple, right? And I think that's something that, especially early on in the conversations with people, they're kind of shocked how simple it is to actually run an experiment in the calScreener. You can just basically go through exactly what it entails, like how you would actually start testing one of these probiotic products. Did you get that, Katrin? Did you hear?
No.
Okay. Just basically, if you could kind of outline the process for testing, what does it actually look like? So say there's no dyes needed, it's non-destructive, but what do you actually do? How do you actually run a test in the calScreener? Because yeah, a lot of people are surprised at how simple it actually is.
So I see the question here. From the present of inactive but still viable cells, if that would complicate the quantification. So what you basically measure is viable and vitable cells, so cells that are directly active, and that is, I think, what you want to have in your product mainly. So this helps you to clearly quantify active bacteria based on the metabolism.
Yeah, I guess for any kind of test like this, you would always also do your own kind of standardization before, so you could really see, like, if there was differences in populations between viable and non-viable and metabolic active, inactive. You would do all of the standards so you could see that quite clearly when you looked at the traces.
Yes.
We have another question from Nancy there about does the instrument allow shaking during incubation?
No, actually it doesn't. So it is, so you add the sample to the vial and then it will be inserted. So it's basically, it grows in batch. What we have tested before, so oxygen isn't a problem for the first hour. So aeration isn't a limiting factor for the first hours of the measurement. So therefore, to assess the viability of your product, this is not a limitation.
Great. Any follow-up questions there? Anything else to add, to ask before we wrap up? Just having a look in the Q&A now. Anything there? Anything in the chat? Okay. It looks like we're good. Katrin, do you have anything that you'd like to add?
No, not at that point.
No. But thank you very much for presenting today. Thank you everyone for attending. This webinar will be available on as a replay, so we'll send you the recording so you can take another look over it. And if you have any questions, like we said, please visit our website, contact us. You can contact Katrin directly or respond to the email that you got about the webinar, and we'd be happy to answer any of your questions. We'll be having more webinars in the future, and we'll be emailing you about those if you'd like to take a look. So thank you very, very much for today, and look forward to interacting with you in some way again in the future. Okay, thank you very much. Goodbye.